So today we're going to be covering one of my favorite topics, and that is the sodium-potassium exchange pump. So just a brief recap of what we were talking about last time. We were discussing the importance of potassium and how it gives rise to our resting membrane potential. Now, in our plasma membrane, we have these things called leak channels, and leak channels are selectively permeable to potassium. Now, because we have lots of potassium inside of the cell, once we open leak channels in our plasma membrane, potassium will want to flow out of the cell. Now, when this occurs, we get a clustering of positive charges, which are potassium ions, around the outside of our cell membrane. And if we remember back to our law of electrical neutrality, for every positive charge we have inside of our cell, we also have a corresponding negative charge, meaning we have an anion. So we get the clustering of anions inside of our cell, and that is the majority of our resting membrane potential. That is the biggest factor. It is the clustering of positive charges on the outside here and the clustering of negative charges on the inside here. However, it is not the only thing that is at play here. So today we're going to be talking about primary active transport in the sodium-potassium exchange pump. So what does that mean, primary active transport? It means that in order to facilitate this exchange, our protein needs energy, and that comes in the form of ATP. So what happens is our sodium-potassium exchange pump draws to it 3 sodium ions. Now, in order to push sodium into the extracellular space against its concentration gradient, we need some help. We need those phosphate bonds that we find in ATP that store all that energy. So because we have adenosine triphosphate, we're able to push sodium into our extracellular space against its concentration gradient. Now, after this has occurred, we get potassium ions then drawn to this little pocket of our sodium-potassium exchange pump. And because we still have a phosphate group attached to our exchange pump, dissociation of this phosphate group then causes a conformational change of our protein, which is a fancy way of just saying it changes shape and then pushes potassium inside of our cell. Now, for resting membrane potential, if we have 3 positive charges leaving our cell and only 2 positive charges coming into our cell, there is going to be an electrical difference created here. And that is the rest of what gives rise to our resting membrane potential. 3 sodium ions out, 2 potassium ions in. Now, this is the rest of what gives rise to our resting membrane potential. Now, it is important that we have these concentration gradients just for normal functioning of our neuronal tissues. Now, if you guys have any questions about this video, please let me know at hubphysiology@gmail.com. For our next video, we're going to be covering the action potential, and that is when we will start to understand why these concentration and electrochemical gradients are so important. All the best, guys. I'll talk to you then. If you have any questions, please let me know. Cheers.